Hello, welcome back to the third week of the Jerusalem Science Contest. This week's subject matter is from chapters 8 and 9. Chapter 8 is plant reproduction, chapter 9 is plant growth, and specifically what I'll be talking to you about today, which is covered a little bit in your textbook, is plant growth regulators, which are known as PGRs. So we'll begin by taking a look at some of the plant growth regulators and talking a little bit about their properties. So let's start with the first slide actually showing you the main classes. As most of these things are actually listed in your text. Auxins, gibberellins, gibberellic acid, what we call abscission agents such as abscisic acid, cytokinins. This one is not, I didn't see this listed, ethylene. And then there are a series of miscellaneous compounds. There actually are some other groups, but most texts will probably tell you that there are 5 principal groups of PGRs, the plant growth regulators. So auxins are probably the best known of these, and they were the first ones to be discovered. They actually frequently act in concert with, or sometimes actually in opposition to other plant hormones. For example, the ratio of auxins to cytokinins, the auxins to cytokinin ratio in certain plants determines the initiation of either root growth, that's downward growth, versus shoot growth. So that's not necessarily made clear in your book. It seems like they're sort of indicating that these things actually act independently, but they don't. At the molecular level, auxins may either stimulate or inhibit expressions of certain sets of genes. Auxins transcriptionally activate 4 different families of what are called early genes, and these are called because the compounds that are actually needed for the gene activation, which is that is to say the auxins, are already present. They don't need to be made. So this results in a rapid response. The early genes, one of the early genes, I should say, is one in which the synthesis of the enzyme glutathione S transferase is involved. There's another one that's called auxin homeostasis protein producing gene, which actually is just controlling the level of auxin that's in the plant. Then there's the SAR gene, S-A-U-R gene, and that stands for small auxin upregulated RNA gene. And finally, the 4th gene that's involved is something called the AUX-IAA, or AUX-indole acetic acid repressor gene. And this last one actually mediates responses to light, how the plant responds to light. On a cellular level, the auxins are essential for cell growth, and they affect both cell division and expansion. Shoots experience axial elongation, so they're going to go up. Roots swell, so they grow down, but they also swell laterally. And fruits undergo isodiametric expansion, which means that a fruit starts real small and it gets bigger in all dimensions. Not all cells make auxins, but auxins are rapidly transported through the phloem or move short distances sometimes by cell-to-cell transport. The direction is usually from the top of a shoot to the bottom of a root. Auxins are involved in all the major tropic responses, which are how a plant would actually respond to certain external stimuli, such as phototropisms, how does the plant respond to light, geotropisms, which is kind of a gravitational response, and hydrotropisms, that is the responding to water. Working in concert with other PGRs, gibberellins being one example, auxins cause cell elongation by stimulating the production of the protein elastin, E-L-A-S-T-I-N, and that makes cell walls looser and more pliable. When you apply to a callus a preparation that has a high auxin to cytokinin concentration, root growth is initiated. So if you've got like a 1-to-1 concentration of auxin and cytokinin, we would call that kind of a neutral state. And then if you get a greater than one auxin, then that's a higher concentration of auxin, or you could have a higher concentration of cytokinin. So when you actually apply that with a higher concentration, then that initiates root growth. However, if you apply a low auxin to cytokinin concentration, then instead of getting root growth, xylem tissue growth is actually initiated. In mature plants, auxins also produce, or rather induce, mineral and sugar accumulation at the site of application. So let's look at the auxins on the next slide. And these are some categories of auxins. So you've got what are called native auxins. These are the naturally occurring auxins, the things that have actually been extracted from plants, and we know that they have this plant growth regulatory effect. All these things are derivatives of this bicyclic two-ring compound, which is called indole. It's got a 6-membered ring here, a benzene ring, and then a 5-membered ring with a nitrogen in it. That ring itself is actually called a pyrazole. Put together, the benzene and the pyrazole make an indole, and CH3, this is a CH2 here, CH3COOH is of course acetic acid, and so this is indole. We actually number from the heteroatom, we number around the ring, so this is the 1 position, the heteroatom being the nitrogen, 2, 3, so the attachment is at the 3 position, and then we would go 4, 5, 6, 7. So this is indole 3 acetic acid, or IAA, and that's mostly when I talk about, unless I mention otherwise, I'm usually referring to IAA as the auxin. Now, there's one here that's a little unusual because it's not an indole derivative, but it is a phenyl, which is the benzene ring itself, again attached to the acetic acid. And that's another naturally occurring growth hormone. But indole 3 acetic acid, IAA, is probably the most potent of these 4 naturally occurring compounds. And then we've got, and you know, by the way, just for your own information, for examination purposes, I don't expect you to memorize all of these structures. However, if I showed you an indole ring, you know, I'd expect you to probably, you know, this kind of a structure right here, I'd expect you to understand that that is indole, and if you saw something like that, you would realize that that is a plant growth regulant of the auxin type. So anything with an indole in it is going to be an auxin. But we've got other compounds that don't have indoles that are synthetic, what are referred to as synthetic auxins. Probably 3 of the 4 that are in here I would not actually categorize as auxins. I mean, they technically are in the sense that they do regulate in some way plant growth, but actually these three compounds, 2,4-D, the compound known as dicamba, and tordon, these compounds are all what I would classify as herbicides. I would call these 1, 2, 3 compounds, I'm sorry, I didn't get the right 3, this one, and this one, I would call those herbicides, and I'll have more to say about them and their herbicidal properties in a later talk. This one, you'll notice, is very, very closely related to 2-phenylacetic acid. This one over here, though, 2,4-D, is something that is commonly used as what is known as a broadleaf herbicide, and it will kill. I know that it's been used, probably use it on my lawn to kill things like dandelions. It doesn't attack grasses, but it does attack broadleaf weeds. So it's a weed killer, basically. Most of the native auxins, however, are compounds that really react the same way in all plants, whereas these synthetic auxins are much more selective, so that they will perhaps kill certain plants, but they won't do anything to other plants. Auxins, the other thing I wanted to say generally about auxins is that, and I think this might actually be mentioned in your book, they stimulate apical dominance and they inhibit axillary growth. It probably doesn't mention it that way, but it does say in your text, I think that if you pinch off an apical bud, that will actually increase axillary growth. That is, that it will actually increase the growth of side branches. And the way that they do this is by initiating the synthesis of one of the other plant growth regulants, ethylene, which is a very simple molecule, in the lateral buds, inhibiting their growth, and that causes apical dominance. If the apex is actually removed from the plant, however, then the inhibitory effect is eliminated and the lateral growth is actually enhanced, which is what we see when we pinch those buds off. Now, in fruit pods, there's a specialized tissue called a valve margin, and this regulates the process of dehiscence. The word dehiscence is D-E-H-I-S-C-E-N-C-E, and that's actually just the opening of the pod. So auxins must be transported out of these margin cells by so-called pin proteins in order for seeds to be released by the pod. And the last thing that I wanted to say about auxins right now is that in low concentrations, they delay flower senescence. So flower senescence being the process in which a flower actually winds up dying. And they do this again by inhibiting ethylene release and transport when the concentration is low. In high concentrations, auxins increase ethylene synthesis, and that also induces female flower formation. I think without the presence of auxins, the flowers would develop naturally as male flowers in plants. Okay, so the next thing that I wanted to talk about are compounds that are known as gibberellins, which, as you can see, are more difficult, at least from a structural point of view. These are more difficult kinds of compounds. These kinds of structures are that, I think I mentioned this before, but not every carbon atom is labeled. So anytime you see an angle here, you understand that at the angle, at that junction, there's a carbon atom. In this case, this is a saturated carbon. Carbon always has 4 bonds attached to it. So here you have a carbon atom with 2 hydrogens, that's 2 of the bonds, and then there's a double bond attached to this carbon atom here. And this carbon atom has 2 bonds, a 3rd, and a 4th bond. So they all have 4 bonds. Now, the one thing that you might not be familiar with is this designation, where you sometimes see some of the bonds will actually be, I don't know how well this actually shows up on your screen, but this is kind of a dark black bond, a triangular bond, and that means that this methyl group, if we look at this as being a planar molecule, so in the plane of the board here, this is this molecule, it's not exactly planar, but if we could sort of flatten it out here, then this group, this methyl group, will be sticking out of the board, be sticking up in the air, along with this hydroxy group, this OH group here, and this carboxylic acid group, the CO2H, is also sticking up. But then you see that there's some other bonds here that actually have a hatch, there's like a little hatch marks in here, and that's indicating that that bond is going in the opposite direction. So not both of these bonds here are sticking up. This one is sticking up, and this bond to the CO here, the bond from here to here, is actually sticking down. So this CO group, carbonyl group, is actually on the other side. It's actually on the back side of the molecule. So these are gibberellins, and there are 4 principal ones, which the first 2 that are shown over here, gibberellic acid 1 and 4, they both have a similar structure. They differ only that in this position, the position shown over here, this group, which is labeled R, in the case of the gibberellin 4, is a hydrogen atom, and in the case of gibberellic acid 1, which is the compound that is shown here, it's actually a hydroxy group. And then we have, I'm sorry, I take that back. This is actually gibberellic acid 3. The difference between 1 and 3 is that this double bond here, this site of unsaturation, does not exist in 1. It's actually a saturated bond, which you can see over here. So this one has 2 hydrogen atoms here, 2 hydrogen atoms here. This one only has 1 hydrogen atom and 1 hydrogen atom. But all diterpenes. They at least start out as a 20 carbon compound. Anyway, they cause cell elongation. They also initiate seed germination, fruit setting, which is actually the process where after the petals fall off of the flower, that a fruit actually begins to form. And they also control both flower and fruit, or they're involved in both flower and fruit maturation. As natural products, the Environmental Protection Agency actually allows gibberellic acid applications to be applied to a farmer's field, and they can be applied at rates of up to about 250 grams, which is a little bit more than 1/2 of a pound per acre. Although there are some environmental groups out there that oppose applications of these things, they actually have very low what are called LD50s, which is the lethal dose that's required to kill 50% of a test animal. In this case, it's most likely the data that I have probably comes from rat studies. But it takes more than 5 grams of these compounds, which is a lot, per kilogram of body weight of rat in order to actually kill a rat. And even at higher doses than that, in human beings, you probably wouldn't see any problem at all. So at a rate of 250 grams per acre, the amount of material that would actually be sprayed on any single plant or group of plants would be minuscule. Anyway, gibberellins have been shown to signal starch hydrolysis through induction of alpha amylase synthesis. Indole acetic acid, by the way, the auxin that we showed on the previous slide, has been shown to regulate the concentration of this compound, gibberellic acid 1, in elongating internodes in pea and tobacco plants. And oxidation of, although this one isn't shown, but gibberellic acid 2 oxidation in barley is decreased from indole acetic acid, while this compound here, GA3 oxidation, that's actually increased. Ethylene, the other PGR that I'll be talking about a little bit later, decreases the concentration of all the gibberellic acids. And finally, gibberellic acids, in concert with abscisic acid, one of the other PGR classes, they actually have an inverse relationship, and they act in tandem with one another to regulate seed development and germination. So abscisic acid, which I mentioned, is on the next slide. And let's take a look at these so-called abscission agents. And there's 2 compounds that I'm showing here. One is the naturally occurring compound, abscisic acid, also called abscissin 2, and also known as dormin. But abscisic acid actually, what it was originally thought to do when it was first discovered, it was thought that its primary function is to induce abscission, or the dropping of leaves and fruit. So there's a layer of cells where the leaf is attached to a plant, and this layer of cells is known as the abscission layer, and these cells would actually die, and the entire leaf, or in other cases the fruit, would actually drop off. It's now known, however, that this compound, it only induces abscission in a small number of plants. Mostly, what I can say about this is also referred to as ABA, abscisic acid. And ABA is produced in the apical buds when the weather turns cold. And this inhibits cells in the vascular cambium, which is a tissue that contains the unspecialized meristematic cells that give rise to cells that actually wind up later on differentiating, and they specialize to form the secondary vascular tissue in plants. So when the weather turns cold, then basically what this is, the function of this thing really is that under these kinds of adverse conditions is you want the plant to stop growing. The plant wants to stop growing at that point. It's going to use its energy in other ways, possibly storing the energy in, it's either going to be making seeds, or if it's the kind of plant that has a tuber or some kind of structure, if it's a perennial type of plant, it's going to be, the energy is actually going to be stored somewhere in the plant, and it's not going to be devoted to actually increasing the size of the plant, because that would be actually detrimental to the plant when the conditions are adverse. So ABA is also produced in the roots, and again, this is in response to things like decreased levels of water in the soil or other kinds of environmental stresses. When that happens, it translocates to the leaves, where it rapidly alters the osmotic pressure in the stomatal guard cells. We looked at guard cells, I think, last week, and it actually causes them to shrink, closing the stomata. So it's kind of a nice feedback mechanism. But when there's not a lot of water, you don't want, the plant does not want to lose water. So when abscisic acid is actually released, what that does is it closes the stomata, and that actually conserves water. Here's this compound called pyrrobactin, and pyrrobactin is a synthetic abscission agent. It also causes stomatal closure. It's an unusual type of compound. It's a this ring, these two benzene rings fused together form the molecule of naphthalene, and this is a naphthalene sulfonamide. It's a substituted naphthalene sulfonamide. It's brominated, and over here it has a pyridine or pyridyl methyl group attached to the to this nitrogen atom here. So this again, and again, I don't expect you to memorize any of these structures, but you should understand that this is abscisic acid. This structure here is the naturally occurring one, and this again is something that was made in the lab, and probably not made, I don't know this for a fact, but I don't believe, I think this was probably synthesized for some other purpose, not as a plant growth regulant, but it was found to have plant growth regulant activity. Years ago, I worked for a company called Diamond Shamrock and was an agricultural chemist, but all the compounds that we made in the agricultural area were also submitted to our pharmaceutical group for screening, and also all the compounds that were made in the pharmaceutical group were submitted for agricultural screening. So we're going to look now at cytokinins, compounds that are shown here. And the first thing that you should notice about these two compounds, kinetin and zeatin, is that these are both derivatives of something that we've seen in DNA, and that is adenine. So this is the adenine part of this amino purine compound is adenine, and adenine, these are both derivatives of adenine, and both of them are naturally occurring, and it's believed that both of these are actually synthesized probably through the breakdown of DNA. I mean, some DNA or maybe RNA winds up getting metabolized to form these compounds. This ring here is called a furan, so this is a furanyl methyl adenine, and this compound here, this ring here has a hydroxy group on it, and there's a double bond in here, and this is an aliphatic 1, 2, 3, 4 carbon, which would be this is a butene, a butenyl group that is actually attached at this position here. So these compounds here, the cytokinins, the first one that was actually isolated and for which the structure was determined was kinetin. It probably actually occurs in all organisms. That's both plants and animals, but it was originally isolated actually from autoclave herring sperm DNA. So it was originally isolated from a fish, and it was thought to be a byproduct of thermal DNA degradation. But it's actually been found. It's been found in other organisms as well. And zeatin is the first part of the name ZEA. Zea, you might recall, Zea maize is the scientific name for corn. So this was isolated from corn. What do cytokinins do? They promote cell growth and differentiation, and they're actually used commercially to increase the yields of fruits and vegetables. They're also used to produce seedless fruits and to increase budding in various herbs. They also affect apical dominance, axillary bud growth, and leaf senescence, other processes that we've talked about. So these are the DNA-derived materials. And the next slide is going to show two fairly simple molecules. This molecule here is just contains a total of 6 atoms, 2 carbon atoms and 4 hydrogens. The C2H4 is called ethene, E-T-H-E-N-E, or the common name is ethylene. And ethylene is a very unusual plant growth regulant because, first of all, it's the only one. First of all, it's the simplest one, and it's also the only one that is actually a gas at anything that approaches ambient temperature. If you wanted to get it to liquefy, you'd have to go down to probably below minus 100 Celsius in order to get this to actually liquefy. So this is a gas. It's released as a gas, and it's very unusual, but it actually, there's a compound here that's called ethafon, which is a synthetic compound, and they're calling it a precursor plant growth regulant. It has plant growth regulant activity as well, but it breaks down, it's metabolized in such a way that it actually winds up losing this chlorine and losing this phosphonic acid group here, and that leaves you with the double bonded situation that you have over here. So ethafon is actually metabolized to give ethylene. So what do these things do? Well, there's 2 genes that are called ETR and CTR, which were identified back in 1993, and they prevent fruit from ripening up until the point when ethylene is actually produced. And what ethylene does is it turns off, it actually shuts down those genes, ETR and CTR. So this causes a cascade effect in which what happens is pectinases, which are, you've heard of pectin, these are proteins that are actually present in the cell wall of many plants, and it actually, the pectinases actually break down the cell walls, and that causes the fruit to soften, which is one of the things that we look for ripening fruit. Of course, if it over-ripens, if it softens too much, then it becomes mushy, and usually it can't be sold at that point. But this actually initiates the pectin release. And then there are other enzymes called amylases, which convert starch to sugars, and then there are enzymes that are called hydrolases, which actually wind up degrading chlorophyll. And as the chlorophyll is broken down, the green color that we associate with plants, particularly in things like leaves, is due to chlorophyll. Once the chlorophyll breaks down, there are actually other coloring agents, other pigments, plant pigments that are present that are obscured by the chlorophyll. So once the chlorophyll breaks down, then we start to see other kinds of colors developing in the fruit, and we associate the turning, for instance, of a tomato from a green color to a red color to the fact that the hydrolases that are present, which are actually initiated by the presence of ethylene, they break down the chlorophyll, and then we can see the underlying color of the tomato. So that's the function of ethylene, short and sweet. And the next thing that I want to show you is a class of compounds that are not mentioned in your book at all, and these are steroidal compounds. The first compound here which actually does not have plant growth regulatory activity, but it's probably present in all plants, is a compound called campesterol. And this is also known as 24-methyl cholesterol. So cholesterol would be exactly like this molecule here, but this CH3 group, which again is, you can see because of the black triangle here, is actually sticking out of the board. And you know, for instance, this hydrogen atom is going into the board because that's got the hatch marks right here. So if I remove the 24 methyl group here and replace it with a hydrogen atom instead of a CH3, I've got a hydrogen here. Then I've got cholesterol. Now cholesterol doesn't really occur except in perhaps trace quantities in plants. Was believed at one point in time that cholesterol did not occur at all in plants. But the amount of cholesterol in plants is actually very, very insignificant. When you realize that if you were to look at all of the lipids, that's the sterols and the oils that are present in plants, per kilogram of dry weight, what you would find in all of the lipids in a plant, you'd find that there'd probably be no more than about maybe 5 milligrams of cholesterol that was present in a kilogram. So that's an extraordinarily small amount, just maybe something on the order of, that would be something like 5 parts per million of cholesterol. If you were to actually look at something like cholesterol in animals, you would find out that the amount of cholesterol there in the lipid tissues, the fatty tissues of animals, would probably be anywhere from maybe 5 to 10 grams per kilogram, and that's 1% of the weight of all the lipids. So literally something on the order of a couple thousand times as much cholesterol. So basically plants do contain a lot of this material, and also the ethyl group, which is a CH2CH3, which is called cytosterol. Remember that when I was an undergraduate, we actually isolated and purified the cytosterol, the 24-ethyl cholesterol, a nice white crystalline compound, which we extracted from peas, from Pisum sativum, which is the common sweet pea that you grow in the garden. So anyway, campesterol, it turns out, this 24-methyl cholesterol is actually metabolized to this other, these, by the way, I think I mentioned before, are actually the tri-terpenes. They start out with 30 carbon atoms. This one actually winds up losing a few carbon atoms. Cholesterol itself only has, I think, 27, so this probably has 28 carbon atoms in it. But it's metabolized to brasinolide, and this ring is expanded here, and there's a couple additional hydroxy groups here. Also, the hydroxy groups actually change their what we call stereochemistry, where the OH group is sticking out. Here, the OH group is sticking back behind the ring. This double bond is removed, and we have inserted here. This is called a lactone. This C double bond O in this ring here. So this is a lactone. So brasinolide is the naturally occurring brasinosteroid, and there's another compound which is a a synthetic compound called abrasin, or also called bikinin. And these compounds, what they actually do naturally is they promote stem elongation and also promote cell division in plants. And this synthetic abrasin is a Arabidopsis GSK3 kinase inhibitor. So that particular enzyme extracted from the Arabidopsis plant, that's a plant that's in the mustard family, that inhibits this kinase, and that actually activates signaling of brasinolide. So these all of these compounds again act in some way as plant growth regulants. Now, all the compounds that I've talked about so far seem to have something to do with increasing the growth of a plant, and there are actually what are called PGR antagonists. These are actually compounds that inhibit actually slow plant growth. So these diverse compounds, very structurally different compounds here, and cimidol, chlorocholine chloride, which is actually close to a natural product. If this had an OH group in here instead of a chlorine here, this would be choline chloride, and choline is what gets acetylated, that is a CH3CO group is attached to it to form what is called acetylcholine, which is one of the nerve transmission molecules. And the last compound here is called paclobutrazol, and paclobutrazol, again, all of these three all act as PGR antagonists, and specifically in this case, they all inhibit gibberellic acid. For instance, chlorocholine chloride, the one shown here, when things like potatoes or other tubers are actually treated with this compound, then the growth of shoots from the tuber is decreased, and the stolons, the growth of these things are actually decreased as well. But in fact, the reason for applying these in an agricultural situation is that they increase the yield of the potato tuber. So you'll get more tubers, and you'll actually get larger tubers per acre. So this is something that is actually sometimes used by farmers. And on the very last slide, I want to show just a couple other miscellaneous compounds. There's just two compounds in here. The first, actually the second one here, fluoroglucinol, is a derivative of phenol, which is just this ring with one hydroxy group. So this is called 1. We number around the ring: 1, 2, 3, 4, 5. This is 1, 3, 5. Trihydroxybenzene, also called fluoroglucinol, and fluoroglucinol, as well as other phenolic compounds. The phenolic would be anything that had the 6-membered ring, and the OH group on it would be a phenol. All of these things act synergistically with indole acetic acid. So if you have IAA and you apply something like fluoroglucinol, you'll actually get a synergistic effect. That will get more of a growth promoting activity than you would from either of these things individually. The other compound here, jasmonic acid, is actually another natural compound. Both of these things occur naturally. This one is very easy to synthesize in the lab. This one's a lot harder, but jasmonic acid, which is this natural product, stimulates the expression of protease inhibitors. And it's actually released by plants in response to insect attack. And it's a powerful natural insecticide. So what it does is it actually prevents insects from obtaining the amino acids that are needed for growth by inhibiting protease, which is actually what breaks down proteins that the insects would be ingesting. It prevents them from getting the amino acids or the smaller polypeptides that they might be able to use in order to synthesize compounds that they require. So the insect, if it's feeding on a plant that has produced jasmonic acid, and most plants will, once the insect starts to feed, that will actually may kill the insect. Of course, it depends on how much they eat. But if they were to eat enough of it, and then there are plants probably insects rather, that probably develop resistance to these kinds of things. But that doesn't refer to its PGR activity. So as a PGR, this is something that actually inhibits plant growth under adverse conditions. So when you get to these kinds of conditions, where you have a limited amount of water, or perhaps the weather turns very, very cold, jasmonic acid is going to be released by the plant, and that will actually slow down plant growth. So that, again, is going to be protective to the plant. Well, that's what I've got to say about PGRs, and again, we'll have our usual 25 question test, 6 questions from this lecture, and again, don't spend too much time maybe just recognizing the basic features of chemical structures that I mentioned, being able to differentiate between some of the different types of structures, but I wouldn't really worry about, certainly not going to show you a structure and say, what is this, or what is this compound? So, and I don't expect you to even know necessarily the names, but you should know indole acetic acids. You should be able to recognize a steroidal kind of compound. Let's go back. So, you should recognize this kind of a ring system indicates a steroid. So a brasinosteroid, anything that would have this kind of a structure or this kind of a structure. It's 2 6-membered rings, a third 6-member ring up here, and a 5-member ring here. Those things are all sterols or steroids, and you should know those kinds of structures. And let's see if there's anything else in here that I really want you to be familiar with. Certainly you should know ethylene, which is just 2 carbon atoms with a double bond between them. And the cytokinins, you should recognize, what I'd like you to know here, get out of this, is these are DNA-derived materials, that they all have this adenine, the part of the molecule that actually is derived from adenine of DNA. So that's what I'd like you to know there. And the abscission agents, I wouldn't worry about the structures here at all. The gibberellins, I think you should probably be able to recognize when you see this kind of a structure, something that looks like that it is a gibberellic acid derivative. And the indole ring as well is something that I think is important. So again, thank you for your attention, and good luck on the exam. And I'll see you next week. Thank you.